tell you anything about kinases, actually. I'm going to tell you about work that I've been doing with Peter Volinis to try to integrate folding landscapes with our understanding of partially folded states in proteins. And in particular, I'm going to concentrate on Ankyrin Repeat proteins, which are the ones that we actually understand the best. And even more in particular, I'm going to tell you about work that we've been doing on IkBa alpha, which is an Ankyrin Repeat protein. And in case you're not familiar with what IkBa alpha does, it actually holds the transcription factor NF-kappa in the cytoplasm. And so it does this by sequestering the nuclear localization signal that's present at the C-terminus of one of the heterodimeric partners of the NF-kappa transcription factor. And in order for this inhibitor to be released from the NF-kappa, it has to be phosphorylated and ubiquitinated on an N-terminal segment that is not seen in the crystal structure. And that targets the complex to degradation by the proteasome, releasing the NF-kappa to then translocate to the nucleus and start transcription of NF-kappa activatable genes. And one of the things that's really interesting about IkBa alpha is that it has very different intracellular half-lives. When it's bound to NF-kappa, it's actually exceedingly stable. It's very hard to measure any degradation of IkBa alpha when it's bound to NF-kappa. On the other hand, newly synthesized IkBa alpha, if it doesn't find an NF-kappa to bind to, it has an intracellular half-life of less than 10 minutes. And this degradation pathway is actually completely different from this degradation pathway. This pathway does not require phosphorylation or ubiquitination. It just happens because this protein is inherently unstable unless it's bound to its binding partner. So this is the crystal structure that was solved simultaneously by Lori Ghosh and also by Steve Harrison's lab. And what you see, the important features of it are, first of all, the blue part is the IkBa alpha, and you can see its repeat architecture here. And this will become important throughout the rest of the talk, how this thing is folded and how it binds. But I just want to point out two other features of this complex structure. One is that there's a very large interface of binding here. About 4,000 square angstroms are buried upon complex formation. And this little pink two-helix part here is where the nuclear localization signal is. It's actually a 4-residue sequence in between these two helices. And then there's a pest sequence at the C-terminus of the IkBa alpha. Okay, so first when we started working on this system, we wanted to understand the binding of IkBa alpha to NF-kappa, and there have been many different estimates in the literature with different types of binding experiments. And we chose to do surface plasmon resonance to understand the binding of these two proteins. And by surface plasmon resonance, you can see that basically once this complex forms, it's almost irreversible. You have to heat the thing up to 37 degrees to actually even measure a dissociation rate in these experiments. And this was actually really nice because the intracellular half-life was being measured simultaneously when we were doing these measurements. And these data fit very well with the measured intracellular half-lives. And I should say that some of the other binding experiments, like gel ships, gave a much weaker binding affinity than this, and those numbers were not consistent with the intracellular half-life. So it really is kind of interesting to see that in these sorts of complex interfaces, different biophysical measurements can give you different binding affinities. But anyway, what we did was we started lopping off pieces of this structure to try to see what was determining the binding affinity. And one of the first really big surprises was that if you just truncated the NF-kappa B P65 component, just take off this little helix here at this end of the interface, you essentially wiped out binding. So this is exactly the same SPR experiment as this one, except we no longer have this little helix on the end of the protein. And the binding is about 8,000-fold weaker. This is just showing you that we actually could recapitulate those results with isothermal titration calorimetry. So this isn't some kind of an SPR artifact for those of you who don't like SPR. Okay, so then at the other end of the interface, we truncated 11 residues just past where you see the interaction here, just from like here on out. And again, the binding was dramatically weakened. And in fact, Guri had done some mutations at interface residues, and he never saw much of an effect on binding. So all these data taken together point to the idea that the binding, the tight binding of this Ankyrin repeat protein to its partner is essentially controlled by two very small regions of the interface, one at one end and the other at the other end. And that the middle whole big swath of surface area is essentially not important for the binding energy at all. So the question then is, can we understand maybe why the binding energy for this complex involves interactions just at the interface, and at the ends of the interface. And so there are some biological reasons. These are the sort of thoughts here that I threw up. One is that we know that proteasome clipping is involved in degradation of IkB alpha, both when it's in complex and when it's free. And so it's possible that you need to make the ends of the interface somewhat like Achilles' heels, if you will, so that the proteasome just has to start degrading and then the entire complex comes apart. Another hypothesis is that it's possible that we actually gain binding energy. Yeah, thank you so much, because that one was done. It's possible that we actually gain binding energy by solidifying contacts between adjacent Ankyrin repeats. In other words, that perhaps the IkB alpha repeat protein is sort of a slinky of sorts, and that when we bind at the ends, we squeeze it, and we actually get folding energy of the inner repeat interaction surfaces. So the question is, can we understand perhaps how the folding of IkB alpha is linked to its binding energy? And the first thing we have to do then is to understand how folded is it when it's free versus bound. And so, well, I was going to introduce Ankyrin repeats just for a second, but I think you don't need to know any of this. All right. So we first did perfectly funneled folding landscape models, or GO simulations, which have already been introduced. Thank you very much to all the people who talked about GO today, Carol and Benoit. So we did a bunch of different Ankyrin repeat proteins, and we just did a bunch of trajectories. Again, what we're looking at here is just an energy function based on the native contacts that are observed in the final crystal structure of the native state, and seeing how those contacts form along a trajectory. And what you can see is that all these small Ankyrin repeat proteins actually show very cooperative folding. And this was somewhat of a surprise at the beginning, because you might imagine that repeat proteins, that you might be able to fold the first repeat first and then the second, or the second repeat first and then the first, and that it would have multiple parallel pathways of folding, and that this might be much less cooperative. But all of these Ankyrin repeat proteins fold in a highly cooperative. These are just CV plots versus temperature, and they're all highly cooperative. So then we had a surprise with IkB alpha. Now what we did with IkB alpha is we took the IkB alpha structure out of that complex that Guri solved, and we said, okay, now for the contacts that we see in the IkB alpha portion, what do we see when we do GO model simulations on that? Okay, and what we saw was actually two folding transitions, and this is the only Ankyrin repeat protein so far that we've seen this with. And so then if you go in and dig in further into each of those folding transitions, in the first folding transition, what we see is contacts forming between the second repeat and the third repeat of I kappa B alpha. And then later on, in the second transition, we see contacts being made in the fourth, fifth, and sixth repeats of I kappa B alpha. And so we postulated that actually what we were observing in these Go models was in fact the transition of a completely unfolded I kappa B alpha to a folded free state in which these contacts down here in the fifth and sixth repeats had not been made yet. And then a second transition from folded free to folded bound. Now remember, the structure that we use in these Go model simulations was the folded bound structure, just taken out of that crystal structure of the complex. And so this was our idea that maybe we were actually capturing both of these folding transitions in the Go models. So we turned to amide hydrogen deuterium exchange experiments to try to see whether that hypothesis had any bearing. And the way we do these experiments is very simple. We just take the native protein, soak it in deuterated buffer under physiological conditions for short times, then quench the exchange reaction, digest the protein, and analyze all the peptides on mass spectrometry. And some regions of I kappa B alpha exchanged almost no deuterium in these short time points that we were using in this experiment, and others exchanged quite a lot. And we were very lucky because this is a repeat protein that we actually got peptides that corresponded to the same regions of each of the repeats. So what I'm going to show you now is just data from these beta hairpin regions of each repeat compared one to the next, because it is a little tricky to analyze hydrogen deuterium exchange data of these peptide regions when they might cover several different secondary structural elements or different parts of the protein. So here's what we got for the free protein, and essentially what we saw was that the second and third repeats are not exchanging almost any amides in the free state. And that, remember, is very consistent with this idea that the contacts are made first in the first folding transition, whereas the first repeat, the fifth, and the sixth repeats are exchanging all of their amides by the very first time point that we can measure. So again, blue signifies low amide exchange, and that is exactly where we saw the high probability of contact formation in the first transition of the Go simulations. So we already kind of had an idea that Ankyrin repeats 5 and 6 were not part of a cooperative folding transition for this protein because we had done a truncation where we expressed only the first 4 Ankyrin repeats. And you can see that there's this sort of pre-transition baseline in this folding curve of the full I kappa B alpha Ankyrin domain. And that is gotten rid of by lopping off that fifth and sixth repeats. And you see the same cooperative folding transition, just missing this pre-transition baseline there. So now I'll tell you what we did with hydrogen deuterium exchange on the complex. And here what we did was we just formed the complex and then did the deuterium exchange and then pulled out the NF kappa B. So we analyzed the HD exchange of I kappa B, but it had been deuterated when it was in complex with NF kappa B. And so what we saw was actually in the first repeat, slightly less deuterium exchange in the complex versus in the free. And this is consistent actually with the surface area change that you can calculate from the crystal structure. The second repeat and the third repeat didn't change at all between complex and free. The fourth changed a little bit, but we saw these massively decreased exchanges of amides in the complex for the fifth and sixth repeats. And there's no way to rationalize 10 or 11 amides difference in exchange here just on the basis of surface area contact. This is clear evidence that these repeats are actually folding on binding to NF kappa B. And that was also very consistent with the Go model simulations, where we saw in the second transition contacts being made in this fifth and sixth repeats. And that's exactly where now, in the folded state, we see protected amides, whereas in the free state, we didn't have protected amides. So now the next question is, can landscape models help us to understand the balance between entropy and enthalpy in this protein folding reaction. And so again, we're going to use these very simple folding models with only native contacts. And here, what we can do, because these repeat proteins are essentially one-dimensional, is, in other words, they have no long-range complex contacts being made. They have all only local contacts. We can actually run lots of simulations and obtain all these trajectories and analyze the number of contacts at each fold-on. And I use this word because we don't really know up front what to call a folding unit in these repeat proteins. They may or may not be repeat as you might see it in a genetic sense. So we're going to sort of slide a window down this thing and try to figure out, are there fold-ons in this repeat protein? In any case, then we can determine the probability of each state at many temperatures and actually write a Hamiltonian to describe the free energy of all the states and to fit these probabilities of states from the simulations. So this is a Hamiltonian. It's really very straightforward, and it's based on an Ising model, which basically says that when a neighboring subunit is folded, there's a favorable interaction energy to fold the one next to it. And again, this is commonly used in these repeat proteins. And then there's this very interesting balance going on here between the fact that an unfolded subunit contributes favorable entropy, whereas a folded subunit contributes favorable internal energy. And so we can see then the full landscape of the folding of these repeat proteins, because their folding again is architecturally simple. So this is one of the landscapes then that Diego Ferrero calculated using this Hamiltonian approach. And one of the interesting surprises that came out of these initial attempts at making real folding landscapes, these are not pretty little pictures, but these are actual energy levels for these intermediate states. So what we found was actually that very small perturbations would have dramatic effects on the stability and folding cooperativity. And you can find many states that are close in energy to the native state at intermediate levels of foldedness. So the reason I bring this up is because we actually then can go in and probe this energy landscape, making single point mutations. And that kind of counterintuitively, despite the fact that you have these many states that are similar in energy, the folding is still actually highly cooperative, and it appears two-state, even though there are many ways to get down that funnel, I guess is a good way to say it. Okay, and so then these landscape models also show broadening of the transition state with sequence inhomogeneity. So we actually were able to do a lot of different CV plots of consensus repeat proteins and to see that their transition states are very narrow, whereas naturally occurring Ankyrin repeat proteins actually have broad transition states due to sequence inhomogeneity. So what we're going to do now is take IkB alpha, which is an inhomogeneous repeat protein, and make its repeats more homogeneous by single point mutations. And so the only mutation I'm going to tell you about, even though we did a lot of them, is one where we restored the sixth repeat to closer to the consensus. We took this tyrosine to a leucine and this, it's supposed to be a leucine, but anyway, to an alanine. Okay, so this double mutation then in the sixth repeat of IkB alpha actually causes the entire protein to be one folded cooperative unit. It was really a surprising result that in the wild type, if you follow, this is a tryptophan in the sixth repeat, and if you follow the tryptophan fluorescence across the folding curve, it basically doesn't change at all. And now if you introduce these two consensus mutations, all of a sudden that tryptophan has this beautiful cooperative transition that follows the cooperative part of the curve. So now this is the entire 6 repeats are in the cooperative unit, whereas here only the first 4 repeats were in the cooperative unit. And hydrogen deuterium exchange also shows that now the 5th and 6th repeats are actually behaving as though they were folded. So, and we took this mutant into cells to actually see if it had a different proteasome degradation rate. This is the free protein being degraded by the proteasome. So this is the 10-minute half-life that the free protein has. And you can see that by stabilizing the Ankyrin Repeat Domain that we actually improved the intracellular half-life by about a factor of 2 or 3 or so. And one of the things that was interesting is that this is a local effect and not a global effect, because we had other stabilizing mutations that didn't show that effect. Okay, but then the surprise came when we actually did protein binding experiments, because you would think that in basic biophysics you learned, I think, that if you pre-fold something, it should bind more tightly because it doesn't have to pay the entropy cost that it had to pay before when it was folding on binding. And we actually got the opposite result, that this pre-folded mutant binds more weakly to NF kappa B. So, and the effect is entirely in the enthalpy. So if you look at the thermodynamics of binding for the wild-type protein, what you see is a very, very favorable delta H for binding, and a very small, slightly unfavorable delta S for binding. And so then the stabilizing mutant actually does invert the delta S for binding. This is minus T delta S, by the way, so you can plot it like this. So it does what you expect it to do with the entropy, but that's actually such a really small effect. And the huge difference is actually that you lose all of this entropy that you used to have, favorable change of entropy upon binding, that now you no longer have. And so we are actually hypothesizing that, in fact, that squeeze mechanism is what's really occurring, and that gives you a lot of favorable delta H on binding, that if you prepay that, you no longer get it after when you're doing the binding interaction. So just to sum up then, that is coming out of here that we actually can now start to really understand, using these really explicit Hamiltonians for writing out real energy landscapes, we can understand the balance between entropy and enthalpy in this folding upon binding reaction. So then, in conclusion, this I kappa B, NF kappa B complex interaction is stabilized by hotspots at the ends of the interface, and actually all of the binding energy is accounted for by those 2 small regions, one at either end of the big interface. The 5th and 6th repeats are only partially folded, but they fold on binding to NF kappa B. And we can make very small perturbations that alter the stability of I kappa B alpha in specific ways, and that will alter the intracellular half-life of free I kappa B alpha. And that really, in order to understand how these sort of metastable, partially folded protein interactions occur, we really need explicit energy functions to understand the balance between entropy and enthalpy in these coupled folding and binding reactions. So I'll just thank the postdocs, Stephanie and Diego, and Simon, who did this work, and Carla Cervantes, who's a grad student. She's doing NMR on this protein, but I didn't present her work today. And this has been a really exciting project that is a program project with Peter Gori-Ghosh, Jane Dyson, and Alex Hoffman. And that's it.
Just a quick question about your point that in your case local stability and not global stability determine this half-life in cells. Isn't that sort of in contradiction to what Andreas Matuszek showed, to sort of this targeted degradation of his model system? Do you have an explanation for that?
Betsy, could you repeat the question?
Okay, so the question is, Marcus Scheck, there's a lot of people out there predicting whether proteasome degradation responds to global stability or local stability. I think in some cases it's going to be, I guess the way I'll answer that question is, if you're doing a global, if you have a globally well-folded protein, then you somehow need to globally unfold it, or you're not going to see proteasome degradation necessarily, right? But this is a case where we already have a marginally stable protein. And so then you can start to really ask the question of, what is the proteasome looking for? Because obviously it's not looking for globular well-folded proteins. But this is one of the really interesting questions that I think we've stumbled upon in this system is, there's this whole subgroup of the Biophysical Society now that's the natively unfolded protein subgroup, right? And actually there's really a continuum between Huntington poly-Q and what we would call, an enzyme, for example. And I think there's just everything in between. And we really need to start to think about, for example, I can tell you this has been a big controversy, but there's no CD change in IkB alpha on binding. And so we now understand that the free state is not really unfolded in the sense that it's a total random coil, that has to form all these helices when it binds. It's almost there. It's really close to being there. Its helices are formed, it only needs those 2 mutations and it's there. So it's really multi-globular maybe, or something very close to being folded, but all its mites are exchanging at the same time. So I think we really need to have these multiple different ways of probing, and honestly, molecular dynamics, when we can get there, is a great way to understand these sort of intermediately folded states, I think. Broader ensembles. And Carolyn, yeah.
So your delta H, delta S, compensation is different directions. So I didn't understand your.
So it's very weak compensation, I guess, is what I would say, right? Because the minus T delta S was unfavorable in the wild type. It becomes slightly favorable. So
Understandable.
Right. But then the thing is, you lose this huge component of your delta H.
So what have you lost?
Well, so that's it. You prepaid it, right? That's the point.
Right.
So the people don't think about this. They don't think about the fact that you can have folding energy in a binding reaction. That's really what's happening.
Maybe the key
it's a
point
folding
is
delta
that
H.
actually those fragments at the end are indeed frustrated. If you look at them, they have lower energy than they should have for folding. That's why we need to make it into a more foldable thing. That's the price that you're paying.
So it's delta H. So it's folding delta H. It's folding delta H within the Ankyrin repeat. It doesn't have anything to do with delta H of contact with the partner. And we don't think about that. Nobody does.
Yeah.